impurity or related substances in the drug product out of specification. This is the topic for the video. You might have observed sometimes that the drug product fails in the related substances or organic impurities. That means the drug product doesn't meet the specifications. In simple way, you can say that the impurities are crossing the limits. For example, if the impurity limit for any impurity, you can say it as a known impurity or unknown impurity, is 0.5%, and you are getting the results like 0.7%, or the drug product is failing in the total impurity limits or in any specific impurity limit. That time the observation will be that the OOS is there. That means the product is failing and the results are out of specification. Out of specification means not complying to the specification limits. This video is related to the impurity specification OOS, or you can say related substances OOS. Out of specification means not meeting the specified criteria, specified limits, or the specification. The out of specification result may be observed during the routine testing. It may be during the stability. But here consider that the out of specification result was observed for the routine testing. The related substances or impurity exceed the regulatory limit. The specification was provided based on the ICH guidelines, based on the pharmacopoeial monograph, or based on the regulatory filings. So what to do? What to do in this case? What are the measures to be taken? What are the steps to be taken? Step 1 is initial laboratory investigation. You have performed the testing and you find the out of specification results for the impurity. Now what to be done? What to do in this case? So first is initial laboratory investigation. To reconfirm testing done using the retained sample. So once you have done the testing, you will be having the samples remaining. So do the testing with retained samples and checks to be performed for the instrument calibration, analytical method validity, and analyst error. And conclusion, no analytical or procedural error was found. You have done the initial investigation for the laboratory testing and you found that there is no analytical error or there is no any procedural error. Then second step is the hypothesis testing. OS was confirmed on retesting. Additional tests included like reference standard, placebo, mobile phase, and conclusion is no lab contamination or interference. See, whenever the out of specification is investigated, it is investigated very thoroughly and in a step-by-step way. So first, initial laboratory investigation is done, and second, the hypothesis testing is done to confirm whether it is actual OS or whether it is a error. The results are because of the error or these are the true error, true results. Then after step 2, step 3 is manufacturing process review. So In this, the review of the batch manufacturing records, PMR, BPR, then deviations, whether any deviation was there, any excursion was there, like change in the mixing time, change in the pH, temperature or hold time was extended or not. So all these risk parameters will be evaluated and studies are done to know that whether there is any issue regarding the manufacturing process, whether any factor is affecting severely for the impurity results or increase in the impurity. Then evaluate the API quality and stability and consider the stress related impurity formation. Go through the force degradation data, go through the stress testing data and confirm the root of impurity formation or the conditions which lead to the impurity formation. Step 4, stability and degradation review. Impurity matched a known degradant. Check for the accelerated degradation, excursion in the storage or transport conditions. This is the example to make you understand. For example, you found that the impurity is matching a known degradant. So you have the data of force degradation, you know the degradant, and now the impurity is matching to the known degradant. So check for the accelerated degradation and excursion in the storage or transport condition, because if the storage is getting changed during the manufacturing, storage or transport, then it may be a reason behind the increased impurity and the OS. Step 5, historical trend analysis. Review the impurity trends from the previous batches. Observe a gradual increase in impurity levels over time. So what your trend data is saying to you, that is very important. The historical trend analysis. So for example, you have observed OS in the 50 number of batch, or you have observed in the 60 number of batch, and before this batch there were already 59 batches manufactured. So that data is required to be reviewed and historical trend is required to be reviewed. So root cause identified, no lab error, and OS impurity is due to process or storage related degradation. Impurity aligns with the non-degradation profile. For example, you find that there is a formation of impurity, it is a degradant, it is a non-degradant, and it is formed because of the storage or high temperature exposure. In first stages, you have identified whether there is any lab error or not. In some next stages, you identified that the degradant is impurity and it is due to the storage related problems or higher temperature during the storage or process. Then go for the corrective and preventive action, because once you find the root cause, you need to correct it and prevent it. So corrective and preventive actions, that is kappa. First point is process optimization to minimize the impurity formation, improved control of storage and environmental conditions, enhanced monitoring of impurity trends in the future batches, and revision of stability protocol as required. These are the very important lines. Once you identify the root cause, you need to do some corrective and preventive actions. You need to do the control points, you need to set some controls. So these are the points to have a effective kappa: process optimization, improved control, enhanced monitoring, and revision of the stability protocol if required. Then regulatory impact assessment, product quality and patient safety are assessed. Risk assessment as per the ICH guidelines. Regulatory notification considered if impurity is genotoxic. New impurity not previously qualified. If it is a new impurity, if it is not matching any degradant, then regulatory impact will be there. Also, if the impurity is genotoxic, if it is found that it is a genotoxic impurity and it is increased, then that batch will not be released. Effective control will be provided and the kappa will be done. OS investigation was systematic and data driven can be the conclusion. Root cause linked to the degradation, not lab error or sampling error. Kappa is implemented to ensure ongoing compliance and the product integrity. So OS was generated or the batch was found with higher impurity level. There was a OS, there was no lab error. So retesting was also done. DMR, BPR were reviewed and it was found that the impurity was a degradant and because of the higher temperature during storage or manufacturing, and then the kappa was taken. So this is the idea for the OS in the impurity and for the kappa. I am thankful to all the viewers that I am getting good response and I am able to let you learn something. I am also learning by making these videos and I am also studying. I request you to provide your suggestions and comments and queries. Thank you.